Hey everyone, it's Chow here, and today we are going to talk a little bit about digestion, so let's get started. So this is just an overview of digestion. I'm not going to be going over specifics. That's something that I might do in the future, or you can most likely look up in your textbook. So let's get started. Okay, so digestion is obviously very important for us as living animals because we need to receive energy and nutrients for proper bodily functions. So that's the key thing about digestion. Now, animals, as animals ourselves, we're not going to be eating constantly. So we must find ways to store nutrients and energy between meals. So we store carbohydrates in the liver and muscle cells as this thing called glycogen. We can also store fat, so adipose tissue, and what's really fascinating is that fat basically has more energy per gram and is much more efficient than carbohydrates because fats are more reduced. So proteins are very important for our body, but we actually don't do that great of a job in terms of storing them. So generally speaking, we're just metabolizing them for last resort purposes. Another thing is that animals require organic molecules to supply those very critical carbon skeletons, which actually can act as building blocks for larger organic molecules. So for example, we have acetyl groups, and acetyl groups, we can think about acetyl-CoA for instance, are used as starting points to build more complex molecules. So if we're trying to build a fatty acid chain, for instance, we need to use those acetyl groups to, bind them together to eventually make fatty acids through a somewhat complicated series of reactions, which I'm not going to discuss. But those carbon skeletons are very critical, and obviously we need to find ways to get those carbon skeletons. Now, in terms of digestion as a whole, what we're talking about is this concept of breakdown, so hydrolysis. Now, in terms of the macromolecules, we talked about in the past lectures that we have to use condensation reactions to build them up. Through condensation reactions, we eventually connect monomeric units to form polymers. So you can think about here maybe like a monosaccharide that's being joined together to make a polysaccharide. Whereas on the other hand, however, digestion is the opposite of that, which is hydrolysis. So we're taking a polymer and through reaction with water, right? So we're basically sticking a water into this bond over here. So if we're talking about carbohydrates, it's a glycosidic bond. We're going to be breaking those bonds to form individual units over here. So we're taking a large molecule and we're reacting it with water, and we're going to break it down into a smaller molecule over here. So there's that. Now, what's really fascinating about digestion is that oftentimes these macromolecules are being broken down by very specific digestive enzymes, also known as hydrolytic enzymes, because obviously they cleave bonds by hydrolysis, as shown over here. Now, these enzymes are very interesting because they're often produced in an inactive form called a zymogen. So the zymogen is the inactive form of the enzyme. And the reason why it's maintained in the zymogen or inactive form is because we don't want those enzymes, which are breaking down macromolecules, to be floating around in their active form in our bodies unless we want them to. But after they are activated by certain environmental factors and internal factors, the enzyme is then free to break down macromolecules. So in the stomach, for instance, we have this zymogen, which is pepsinogen, and the pepsinogen is the inactive form. But when we eat food and then suddenly there's a release of hydrochloric acid in our stomach, that is actually going to cause the pepsinogen to cleave into pepsin, and pepsin is the active form, and that can start slicing apart protein peptide bonds. So zymogen forms, inactive forms, can be activated into active forms, but enzymes in general for digestion are maintained in these inactive forms to basically prevent the body from breaking down, accidentally breaking down things they're not supposed to. Okay, so macromolecule digestion is you're taking a big old macromolecule and you're breaking it down by specific digestive enzymes to smaller monomeric units. So we have many different groups of enzymes to help with this process. The first group are your proteases and peptidases, and these target the bonds of protein. So they break down those protein bonds to form those amino acids. So we're breaking peptide bonds and slicing those polymers into monomers called amino acids. Or if you want to think about it, it's breaking a polypeptide into an amino acid. We also have carbohydrates and glycosidases. These target the bonds of polysaccharides, so your glycosidic bonds, to get simple sugar. So you're taking a polysaccharide and turning it into a monosaccharide like glucose. You also have lipases, and these break down these basically break down fats, so triglycerides, into fatty acid components. And then finally, we have nucleases, which basically break bonds of nucleic acids to get your nucleotides and bases. So some different groups of enzymes that are useful in digestion. Okay, so the process of digestion is actually quite complicated, and it varies between the macromolecule that you're talking about. But the first type of digestion really is mechanical digestion, which is chewing. When we talk about digestion, usually we're talking about chemical digestion, but there's also mechanical digestion, which is simply chewing within our mouths. This doesn't really do that much. It breaks the large food particles to smaller food particles, but in terms of the chemistry, oftentimes it doesn't really change. But we do see a little bit of chemical digestion in the mouth, more specifically with sugar. So we have certain enzymes in our mouths, for instance in saliva, such as salivary amylase, that will break down polysaccharides into smaller, at least disaccharide units. We're not really going to break the polysaccharides in our mouths down to monosaccharide units. That really happens in the small intestine, but we can break down those large polypeptides into disaccharide units. As for lipids, chewing doesn't really do much, and proteins, chewing really doesn't do much either. So you can break down a bigger glob of fat into a smaller glob of fat, but it doesn't really help out all that much in terms of chemistry. In the stomach, that's where we often see some of the initial breakdowns for proteins. So we often see breakdown of proteins by our friend pepsin, which is again the activated form of pepsinogen. We have the gastric mucosa, which is the mucus-secreting epithelial cells in the stomach. We also have parietal cells in the stomach that secrete your HCl, your hydrochloric acid, and that's what's going to activate, right? So that's what's going to activate your pepsinogen into pepsin. So speaking of pepsinogen, we have the zymogen pepsinogen, which is released by chief cells. And then if the environment has a really low pH, and there's a high presence of HCl, we're going to cleave pepsinogen into the active form pepsin, and that can start breaking down that protein polypeptide into smaller polypeptide units. Okay, so then the stuff in our stomach moves on to the next stage, which is the small intestine. And in the small intestine, that's the site of the majority of digestion and absorption. So the majority of digestion and absorption actually happens in the small intestine, not really the stomach. So the small intestine consists of two accessory organs that help provide specialized enzymes for small intestine digestion. The first of these accessory organs is actually the liver and the gallbladder. And so the liver and gallbladder actually work together. And so the liver actually produces the bile, and then the gallbladder stores it. And the bile, which is released, acts as an emulsifier of fat. So it actually breaks fat globules back down into smaller droplets. So you're breaking down big old fat globules into smaller fat globs, and that can help, right? That can help out with digestion. We then also have lipases, amylases, proteases, nucleases, and other different types of enzymes that are going to be released by the second accessory organ, which is really important, and that's the pancreas. So the pancreas releases all these different types of enzymes to basically help break down the peptides into smaller units. So really, it's in the small intestine, more specifically in the duodenum, that the pancreas is going to be releasing these enzymes, and those enzymes are going to be what's actually breaking down your polypeptides into your amino acids, breaking down those polysaccharides into monomeric units. So this is where all the breakdown happens. And then finally, the nutrients are going to be absorbed through these small intestine walls, which actually have these folds called villi and microvilli that actually increase this intestinal surface area for absorption. And then finally, there are blood vessels underneath the villi and microvilli that allow for absorption of nutrients. So in the small intestine, we see a release of different enzymes to basically break down all these macromolecules into monomeric units with the help of other secondary things like brush border enzymes, and they're absorbed into the body. Now here's where it gets a little bit different, because for amino acids as well as monosaccharides, they're actually going to be absorbed directly into the bloodstream, and then they're going to pass into the hepatic portal system up into your liver. Whereas with fats, the fats actually can sometimes have proteins added onto them to make these chylomicrons, and they actually are put into the lacteals and actually run through the lymphatic system before they actually enter into your blood. So that's a little bit of a difference between the absorption levels. I don't know if they're going to ask you for that level of specificity, but it's definitely something to keep in mind and maybe read over in your textbook. So there we go. Okay, what else do we have about digestion? Okay. So after the small intestine, the particles that are left basically enter into the large intestine, also known as our colon. And the colon allows for a bit of absorption of water and ions, and it's going to store the undigested waste as the feces. And then eventually it reaches our rectum and then our anus, and that also allows for temporary storage of feces until we, have to get rid of them. Okay, so the last thing that I'm going to discuss is, what about that glucose? Why is it that in terms of digestion, why is glucose such an important thing? Why is it that we always talk about this stupid thing called glucose? And the reason why is because, well, glucose is quite important for our bodies. And our bodies actually maintain this very important energy source called glucose from food, especially because of our brain. Our brain actually primarily uses glucose and not really much else for energy purposes. So we have to end up regulating glucose levels within our body. And one of the organs that actually helps basically regulate these glucose levels is this one over here. Again, the pancreas. So it monitors the set point for glucose levels, and it checks for glucose levels that fall below normal or rise above normal. And so we have different hormones that are secreted by the pancreas that can help with this process. One of them is one that you probably have heard of, and that's insulin. So insulin actually is released when glucose levels are high in the blood. And the release of insulin triggers the uptake of glucose by cells. This leads to metabolic energy production, for instance, fat synthesis, as well as glycogen synthesis. So obviously, if you are diabetic and you have some kind of an issue with a lack of insulin, you might have high blood glucose levels, but you're just not being able to absorb them into your body. And the insulin actually causes a lot of interesting cascades, which we're not going to talk about, but it can help regulate various levels of glucose as well as other indirect things within the body. We also have the release of glucagon, and glucagon secreted by the pancreas does something quite similar to epinephrine in that it induces the breakdown of glycogen in the liver into glucose, and it ultimately releases that glucose into the blood for use, and obviously it increases the circulating glucose levels. So if we're looking at these systems almost like feedback systems, you can see that in the fed state, insulin is going to generally be dominating. So insulin is going to be released when you are really full, and that can help increase levels of glucose oxidation, increase levels of glycogen synthesis, fat synthesis, as well as protein synthesis. Whereas on the other hand, if you're fasting or if you're starving or hungry, you're most likely going to see an increase perhaps of glucagon. And so glucagon domination will cause glycogenolysis, so breakdown of glycogen, as well as gluconeogenesis, which is generation of glucose, and then also the creation of ketone bodies through this process of ketogenesis. So that's basically all I have in terms of digestion. We went through some of the basics of digestion as well as why glucose is important for the system. So please read over the textbook, read over what's expected by your professor, and I hope you found this useful. And as always, best of luck studying, and I will see you in the next video. Thanks, everyone.